Morning to all. I am Dr. C. Arunmati, Assistant Professor, Department of Biochemistry, J.J. College of Arts and Science, Pudukkottai. Today we are going to learn about the topic lymphoid organs. Before we enter into the topic lymphoid organs, we must know about what is about the immune system. Our environment consists of numerous pathogens that includes bacteria, protists, fungi and other infectious organisms that are able to cause various diseases. The defense system that is capable of providing protection from the majority of these disease agents. Our immune system is occur in multicellular organisms as in a simple form, for example, in ancient form as sea sponges. Our mammalian system is made up of both individual cells and proteins as well as the entire organs and organ systems, which are known as the lymphoid system. This immune system takes a specific action for neutralizing, killing and eliminating the agent. This immune system includes skin, mucous membranes are the first line of defense against the germs they are entering from outside of the body. The antibacterial substances which can kill the germs, for example, there are certain enzymes that are found in saliva and tear. That fluid destroys the cell walls of bacteria. Next, the mucus which is present in bronchi helps to trap many of the germs by its hair-like structures called cilia. Like that, stomach acid that stops most of the germs that enter the body in the food that we eat. Now we see about the major classifications of the lymphoid organs, that is subdivided into primary lymphoid organs and the secondary lymphoid organs or tissues. First we see about the primary lymphoid organs. They are subdivided into thymus and bone marrow. Next we see about the secondary lymphoid organs, spleen, lymph nodes. These two are the organs, and the tissues like mucosal associated lymphoid tissues. Next we see about bone marrow. It is a soft gelatinous tissues that present in the central cavity of long bones such as femur and humerus. The blood cells and immune cells which arise from this bone marrow, and they develop from immature stem cells that is known as hemocytoblast, and then they follow the distinct developmental pathway to become either erythrocytes, leukocytes or platelets. This process is known as hematopoiesis, that is the synthesis of heme. And then this hematopoietic stem cells that include myeloid progenitor cells, which include monocytes, macrophage, neutrophils, basophils and etc. And like lymphoid progenitor cells like T lymphocytes, B lymphocytes and natural killer cells. At birth, the whole skeleton is filled with red marrow, but at puberty it is reached, the growth is slowed down and the physiological conversion occurs at that time, and changing red bone marrow into the yellow bone marrow. The red bone marrow in the axial skeleton and articular ends of the humeral and femoral bones. Yellow bone marrow means it is present in the most distal parts of the skeleton. The bone marrow diseases, that is the effect of any defect in the bone marrow or bone marrow diseases like leukemia, Hodgkin's lymphoma and other lymphomas. And next we see about the Bursa of Fibrosis. It is the primary lymphoid organ of birds. Its main function is hematopoiesis and the development of B lymphocytes, which like in the mammals. It is located at the dorsal to proctodic region of the cloaca and has the form of a single bag. The surface of bursa is constituted by folds that give its appearance like a book sheets. The folds have follicles that containing follicles associated epithelial cells, lymphocytes, macrophages and plasma cells. Its normal size is equivalent to the size of the spleen in healthy birds of 2 months, but it may be diminished by the action of pathogens. It is active in young birds, but it begins its atrophy, that means the shrinkage of this organ after about 6 months. Next we see about the major lymphoid organ in thymus. It is a bilobed pinkish grey organ located just above the heart in the mediasternum, and it is rests below the sternum, that is the breastbone. It is found at the breastbone. In prepubescence, it is a relatively large and very active organ, and it weighs about 40 grams. But it in a middle age, that is in adult age, it may have the shrink. It is known as atrophy, and it's sufficiently to be difficult to locate it. In the sixth of its original size is shrinkage, that is known as thymic involution. Each of these two lobes of the thymus, it is surrounded by a capsule within which are numerous small lobules. Size is about 2 to 3 millimeter in width, which are held together by loose connective tissues. Each lobules consist of the follicles that are composed of a network of framework of thymosin hormone, that is secreting the epithelial cells of T lymphocytes, and that are commonly referred as T cells. Next we see about the lobules. The lobules have a dense outer cortex that is present in outer cortex and rich in actively dividing T cells. In its inner medulla, it is much paler in color and functions as an area of T cell maturation. And that's its activation or programming of T cells, that is to recognize as self-antigens through by its thymic education. Mature T cells, that is continually migrate from the thymus into the blood and other lymphoid organs, that is from spleen and lymph nodes, where they play a major role in body-specific immune responses. And see about the disorder of thymus, that is thymic cancer and myasthenia gravis. And next we see about the secondary lymphoid organ, major organ, that is the spleen. It have a key role of secondary lymphoid organ, and its main role is filtering out and destroying the unwanted pathogens. The maintaining the population of mature lymphocytes, that is the white blood cells, to enable the adaptive immune response when they begin. This largest lymphoid organ, it is situated in the upper left of hypochondriac region of the abdominal cavity between the diaphragm and the fundus of the stomach. It is like a dark purple oval shaped organ, and it is enclosed in a dense fibroelastic capsule that protrudes into the organ as a trabeculae. It is made up of two regions. First one is stroma, that is comprising the denser outer capsule with its trabeculae, some fibers and fibroblast, that is the cells that secrete connective tissue of collagen. And second one is the parenchyma. It is composed of two types of intermingling tissues called the white pulp and the red pulp. And the major functions of spleen: to mount an immune response and remove the microorganisms from the circulation, and to destroy damage and the worn out of red blood cells, to store the platelets and sometimes the blood. The enlargement is known as splenomegaly. And we see about the next secondary lymphoid organ, lymph nodes. These lymph nodes vary in size and shape, but they are typically bean-shaped structures found clustered at specific locations throughout the body. Fibrous capsule. These forms have a protective outer sheath and has a trabeculae that extend periodically into the node and then subdividing into small compartments. Then the outer cortex or the nodular cortex. It is present just inside the capsular margin. This consists of numerous follicles that are rich in B cells. When the pathogens are present, these follicles expand to reveal prominent germinal centers containing actively dividing antibody, the secreting B cells. Next, the inner cortex, that is known as paracortex. It is found just below the outer cortex and is particularly rich in T cells. They also continually circulated throughout most of the regions of the node. Then the medulla, the central inner portion of node that contains large number of fixed phagocytic macrophages, and they continually monitor the lymph for potentially pathogenic foreign material. This process is known as immunosurveillance, and that they phagocytase on the contact. During that infection, the antibody producing B cells began to proliferate, that is, multiply in the germinal centers, that causing the affected lymph nodes to enlarge and become palpable and tender. Some of the cytokines, they are released from pyrogenic, that is, they cause the fever, and they act directly on the thermoregulatory center, that is the hypothalamus, and thus increase the body temperature. Swollen lymph nodes and a fever, that are both sure signs of that body that is mounting an effective immune response against the offending pathogen. And next we see about the mold. It contains small aggregations of lymphocytes called lymphoid follicles that position to protect the respiratory, urinary tract and gastrointestinal tracts from the invasion by microbes. The following are made up of the small tissues. First one we see about the nasal associated lymphoid tissues as NALT. The two lymph nodes that are located on both sides of the back of throat and nasal passage with immune cells. The tonsils that are aggregates of lymphatic tissues and strategically located to prevent the foreign material pathogens from entering the body. Then the palatine tonsils, they are present in the pharynx, the lingual tonsils present in the oral cavity, and the pharyngeal tonsils that are known as adenoids and are at the back of the nasal cavity. And as a result of this, the tonsils themselves have the high risk of infection and inflammation, that is known as tonsillitis. Next we see about the gut associated lymphoid tissues as GALT. It is composed of lymphoid complexes and consists of specialized epithelium, antigen presenting cells and intraepithelial lymphocytes. These structures occur strategically at specific areas in the digestive tract, for example, Peyer's patches in the terminal ileum. These specialized cells are called as M cells, which are intimately associated with antigen presenting cells and together carry out antigen sampling in the gut. Next one, Branchus associated lymphoid tissues or BALT. These are structurally similar to Peyer's patches and other lymphoid tissues in the gut. It is composed mainly of lymphocyte aggregates and organized into follicles which are found in all lobes of the lungs and along in the main branchial. The majority of lymphocytes in the follicles are B cells. The antigen sampling is carried out by this epithelial cells lining the surface of the mucosa and by way of M cells that transport antigens to underlying antigen presenting cells and lymphocytes.